viruses replicate? So we said they need a host, but then what happens inside the host? How does replication occur? How do they make more viruses and do that? So let's look at an example of replication that's been very well researched and characterized, and this is called uh, this is a bacteriophage method of replication, and there's two ways that this happens, the lytic cycle and the lysogenic cycle. So again, this is a bacteriophage. What's the definition of bacteriophage? Viruses that infect bacteria. And again, you may be wondering, why do we care? We want to talk about viruses that lead to diseases in humans. Why are we learning about viruses that infect bacteria? Well, the more we learn about it, the better we can protect ourselves. Learning about viruses in a simple structure invading a bacterium can help us when we're trying to learn it in a more complicated eukaryotic cell. So again, learning something simple and then applying that to eukaryotic cells and viruses that infect eukaryotic cells, such as ours. So this is bacteriophages. Bacteriophages are viruses that attack bacteria, and we see that they reproduce in two different ways, the lytic cycle and the lysogenic cycle. Now, before we learn about this, I want to show you, these are some size pictures that make it crazy. All right, so you remember bacteria, right? And again, think about it, 1,000 times magnified, and you get little bitty circles or little bitty ovals. This is an electron microscope image, so we've magnified even more. And these circles right here are virions. These are viruses that are not inside a cell yet, so they're called a virion. A virion is a virus outside of a host. So that's the definition of virion. And what they're attaching to, this little line through here, is a pili. Do you remember that? That's that specialized fimbriae that attach to bacteria cells. So again, imagine 1,000 times magnified, and with those little circles you were sawing, those are the bacteria. We can't even visualize a pili, but those are the viruses attacking it. This is one bacteriophage, and it's attached to a flagellum. So again, flagellum, we're going to do staining this week, and flagellum staining is so difficult. We don't actually do that in the lab. It's because these flagella are so fragile. They're very thin. And here's a picture of a bacteriophage attached to one of those. We'll come back and look at this picture in a minute. But again, wrapping your head around the size difference that we're talking about here. Okay, so let's talk about these replication cycles. The first one we're going to talk about is referred to as the lytic replication cycle. Some bacteriophage replicate by the lytic cycle of replication. Now again, how they replicate, they have instructions in their DNA or in their nucleic acid on how to do this. So if a virus is going to replicate by the lytic replication stage or the lytic cycle, then it's always going to do that. Okay, so keep that in mind. So let's go through the steps of replication and figure out how this works. Okay, number one, in order for a virus to get into a host cell, it's got to find that host cell, and that's by chance. It's not seeking it out, but if it happens to come in contact with a host cell, remember it has those glycoproteins on the outside of the virion of the surface, that will attach to something on the cell. The cell also has proteins, right? Phospholipid bilayer, integral proteins, peripheral proteins. So some protein that the cell has, that the cell uses to do whatever, has become a target for this virus, a way that it can get inside the cell. So the first thing that has to happen is a virus has to attach to a host. Chance collision, because they can't move on their own. So number one, attachment. Once it attaches, and again, that's protein-protein attachment, a glycoprotein spike on a virus with a receptor, which is just a regular protein on the host that the virus is able to attach to, then it needs to get inside the host. So how does it do that? Well, the virus attaches, and it actually has inside the virion, it has a enzyme called lysozyme. So it has a protein that's packaged inside the virion, and that protein is lysozyme. And lysozyme is a protein that weakens the cell wall. So if it weakens the cell wall, it's going to cause, like it's going to poke holes in the cell wall. It makes it weaker, that peptoglycan cell wall, and that's going to help entry. There's also a chemical reaction, and I think, let me see if I have a picture of it. That one. We'll go back to this picture. It also is going to cause a chemical reaction. So here is a bacteriophage, and it's attached to a protein on the flagellum. It's going to cause a chemical reaction that's going to cause a contraction of this helical tail portion. And that's going to cause the nucleic acid in this head to get injected into the cell. I'm going to have to go to my whiteboard for that. So let's draw a cell membrane. So that's my bacteria cell, and I have a bacteriophage that's going to attach here, and it's got those tail fibers, and it is attached to a protein that's on that. So the tail fibers attached to this thing. This causes a contraction of this helical tail section, and then my nucleic acid that's inside ends up getting injected into my cell. So what actually goes into the cell? It's only the nucleic acid that goes into the cell. The capsid portion remains outside the cell. So attachment and then entry causes this contraction of this, which injects this in. It's only the nucleic acid that enters the cell. So back to my picture that I told you I'm going to come back to. The capsid that remains on the outside becomes what they call a ghost phage. It's not on that one. So this capsid right here, so you can see the head section that has the nucleic inside, and that you can see that nucleic inside fluorescing in this electron microscope image. And then you see the contraction of this tail portion, and you can actually see that green nucleic acid getting injected inside. Now this particular one, the nucleic acid has already been injected inside. This one's in the process. So you can see that it leaves behind an empty capsid. It has no more nucleic acid inside. And this empty capsid left on the surface of the bacterium is called a ghost phage. So I like this picture because it really sees that. You can see that nucleic acid. You can see it actually entering the cell. So first thing, attachment. Second thing, entry. Lysozyme helps that process because it weakens the cell wall and it leaves that ghost phage behind. Well, then what happens? Well, then the virus is going to go to work. Remember, it's got instructions on taking over the cell. It's going to make the bacteria cell make viral DNA. It's going to copy viral DNA. It's going to stop copying its own DNA, and it's going to copy viral DNA. It's going to make the cell ribosomes make viral proteins instead of making bacterial proteins. It takes over. What proteins is it making? All those glycoproteins, those spikes. It's going to make capsomeres from that. It's also going to make some enzymes. It's going to make that lysozyme that needs to be made. It's also going to make an enzyme called nuclease. And nuclease actually degrades bacterial DNA. So this virus convinces the bacteria to make an enzyme that's going to degrade the bacteria's own DNA. It won't degrade viral DNA, but nuclease targets bacterial DNA. So this stage is called synthesis. So that's what's going on in this stage. It's making all the parts and pieces that are needed to make that. Nuclease destroys bacterial DNA. Then what happens? We have attachment, we have entry, then we have synthesis. It's making all the parts and pieces. Then we have some instructions on how to put those parts and pieces together. So chemical reactions occur. We still don't know exactly how that works, but basically the capsid gets made, the nucleic acid, the viral nucleic acid gets packaged within that capsid, so we get assembly inside the bacteria cell. So now I have all of these virions inside the bacteria cell, and eventually the last stage is lysis. Once a virion has entered a bacteria cell, that bacteria cell is going to die. It is going to eventually break apart, lyse, and die. Remember, it was making that lysozyme. Remember, the purpose of lysozyme breaks down bacteria cell wall. So after all this lysozyme is made, it degrades the bacteria cell wall. Now this bacteria cell just falls apart. It lyses. Water is able to rush in and the cell breaks apart. So now the virions escape into the environment. All right, so I want to do a couple more. I want to do another vocabulary time. Like, how long does this process take? How long does it take to go from penetration, entry, to release? And this can be, depends on the virus, but it can be as fast as every 20 to 40 minutes from that. Okay, so that is very quick. We have a chart here, so we can chart this time. So here's time 0. On this axis, we have the number of infected virions. So we're asking how many virions are in the environment, and we start off with 1. Okay, so again, you came in contact with 1 virion in the environment. Either it was a respiratory droplet, you inhaled it, or you touched it and then you scratched your eyes and it got inside your body. You have come in contact with 1 virion and it's gotten inside your cell, inside your body. It moves by currents and it happens to come in contact with a host cell, a cell that it can invade. What happens? So looking at this chart, we talked about what happens: entry, attachment, entry, synthesis, assembly, and release. Time-wise, if we were to plot this out, so here's my number of infective virions in the environment. So here's 1. It comes in contact with a host cell, so it enters. So now we have gone from 1 in the environment to 0 in the environment. At after 5 minutes, it comes in contact with a host cell. Now it enters the host cell, and so now I have 0 infective virions in my environment because that 1 I did have has entered a host cell. So time is passing. We said anywhere between 20 to 40 minutes, depending on the type of virus it is. Inside, we're getting synthesis and assembly. It's taking over that cell. Time is passing. Then we get to the last stage, which is that release by lysis. It has made a whole bunch of virions, and all of those are released when that cell lyses and falls apart. How many can be released? This is called is this on this one? The burst size. So your burst time is how long it goes from penetration to release. Burst size is how many virions get made and released into the environment. And this can be anywhere between 50 to 200. Again, depending on the type of virus and depending on the type of cell that it invades, it can be anywhere from 50 to 200. So you just went from 1 to 200 in half an hour. So now, what are those 200 virions going to do? Well, they're there. They're not looking for anything, but they're there. They're being carried by currents. And if there's 1 bacteria cell in the environment, what's the chance that there's going to be other bacteria cells in the environment of that same type? Really good. We just talked about chapter 6. We talked about binary fission and how quickly bacteria can replicate. You have lots of bacteria in one place. Now I have 200 virions that have been released where there's a whole bunch of E. coli, and they're going to potentially find 200 E. coli cells to invade. And then that happens, and each one of those 200 can make 200 virions, and those get released in another half hour. And then that 200, or 200 times 200, so we do logarithmic, crazy growth. When we talked about that logarithmic growth curve and how quickly bacteria cells can replicate, this type of replication is even bigger because you're not going from 1 to 2, 2 to 4. You're going from 1 to 200. And then each of those 200 go to 200. So 200 to 200 squared. I mean, it's crazy math. I can't even. This is how viruses can so quickly take over from that. All right, so vocabulary term: burst time, the time it takes to go from penetration to release. Burst size, how many virions can do that. Okay, so here is a picture. I want you to stop and I want you to go through with these pictures and define each of those stages. All right, how'd you do? It's easier with a picture. So attachment, cell membrane, you get that. You got attachment, then you have entry, and again, we're studying the Lytic cycle of a specific bacteriophage that invades E. coli, and you get this contraction and ejection of nucleic acid that goes in, leaving the ghost phage behind. We get the phage DNA, it's making nuclease, so we get the bacterial chromosome degrading. We have synthesis, and assembly occurring, and then we have release by lysis. Okay, how'd you do?